In this video, we are going to be using manipulatives to try to understand the relationship between blood type as well as the kind of antibodies a person produces. Here we have a red blood cell, and these little packs we have actually represent the antigens you will find on the surface of red cell. So you can see here the blue pack is representing the A antigen. We have the yellow pack here representing the B antigen. And for the green packs here, that represents the D antigen or the Rh antigen. So let's try to build different types of blood type. Let's start with A positive. When a person has A positive blood, it means that they are going to express the A antigen, represented by the blue packs here, on the surface of the red cell. And also, because they're positive, it means they have the Rh antigen on them as well. So here, the red cell has the A and the Rh. That makes it A positive. So why don't you pause the video for a second and think of how you would build a red cell that is AB negative. After you try that, hopefully you get the same result as me right now. So AB negative means it has the A antigen, which is once again represented by the blue packs, and it also expresses the B antigen, but the person is negative, meaning they are not going to have Rh on the surface of the red cell. And so AB negative would look like that. Now let's take a look at the kind of antibodies each of these individuals will produce. The general rule about antibodies is that your body is capable of producing antibodies for antigens that you do not express. For example, if a person is A positive, then they have anti-B antibodies, which are represented by these yellow Y-shaped things. On the other hand, for someone who's AB negative, okay, they are going to be producing anti-Rh antibodies. Now, for the purpose of our course, a person pretty much is going to be born with anti-A or anti-B antibodies. But if a person has negative Rh, they will only be able to produce the anti-Rh antibodies once they have been sensitized, okay? Meaning once they have been exposed to Rh positive blood. Right? So there you have it, the kind of antigens you have on the surface of the cell and the kind of antibodies that you will have in the plasma of the blood. Next, we're going to look at a few transfusion examples and see what's compatible and what's not. Let's start with our first example here. Suppose we have a donor with A negative blood giving RBC to an AB positive person. Remember, when we're talking about transfusion in this course, we are only focusing on the transfusion of red blood cells. We are not going to be doing whole blood. We're not going to be doing the plasma. So only the red cells will be given to the recipient. Okay, so let's take a look at the donor here. The donor is A negative, and there I have prepared the model for it. Okay, A negative. We're not going to worry about the antibodies because only the red cells will be given to the recipient. And our recipient here is AB positive, so there will be A antigen, B antigen, as well as the Rh on this person. So we have to answer 3 questions when we try to determine whether or not a transfusion is compatible. Number 1, we have to look at the kind of antigens that are on the red cells of the donor. Number 2, we have to look at the antibodies that are produced by the recipient. And we want to see whether the antibodies in the recipient's body is going to attack the incoming red cells. In this particular example, because our recipient is AB positive, they're not going to make any antibodies. They're not going to have the anti-A antibodies. If they do, then what's going to happen is they're going to end up attacking their own cells, right? We don't want that. And similarly, they're not going to have the yellow or green antibodies either. So for the purpose of A, B, O, and Rh transfusion, there are no antibodies in the plasma of the recipient. So when we go ahead and do the transfusion, the red cells will be accepted with no issue. Let's take a look at another example. Let's say we have O positive as our donor, and we are going to be giving it to an A negative person. O positive right here only has the Rh, and for someone who's A negative, it is only going to have the A antigen. Okay, so what kind of antibodies will the recipient have in their plasma? Well, there's going to be the yellow antibodies, the anti-B antibody, as well as the anti-Rh antibody. Okay? Remember, technically, you're not going to have the anti-Rh antibody until you are going to be sensitized. So for our discussion here, let's just assume the person has previously been sensitized and that the Rh antibodies are there. So if we go ahead with this transfusion, okay, then the anti-Rh antibodies in the recipient will now bind to the incoming cell, and that is going to cause a cross reaction, and the cross reaction will cause the red cells to basically burst. Okay, we call that hemolysis, and that is not a safe transfusion. Right. Let's try a third example. Let's say we have the A positive donor trying to give blood to a B positive person. So A positive would look like that. Okay, the blue is the A and the green is the Rh. And here we have the recipient, which is the B positive. Okay, B positive. So what kind of antibodies will the recipient be producing, and whether or not this transfusion is safe? Please pause the video and think about it before continuing watching. So this recipient is B positive, which means they're going to be producing the blue antibodies, the anti-A antibodies. You're only going to be making antibodies for things that you do not have, right? And so if we go ahead and proceed with this transfusion, we are going to have a cross reaction between the antibodies in the recipient and the antigen in the incoming cells. And again, this is called a cross reaction. It will result in bursting of the red cells, okay? And therefore, this is not a safe transfusion. That's all for now. Thank you.